Good morning. I am Dr. Maisa, professor and the head of medical parasitology department. Today we are going to take the second lecture of medical parasitology, which is about classification of parasites. You can see here numerous parasites. Some are worms, and some are unicellular, and some are arthropods. So, parasitic organisms can be classified according to their shape. If they are multicellular, like this picture, it is a large worm. Can you see here its length? It can reach up to 10 centimeter or up to 25 centimeter. It is an ascaris worm. We call it helminthes. They are multicellular parasites. Usually they are bilaterally symmetrical and can be either in the form of a round worm or a flat worm or a segmented worm, or are helminthes, which are multicellular organisms. You can find some of them hermaphrodite, means that they have male and female genital system, or they can have separate sex, like this picture. It is a male ascaris worm. If you remember in the first lecture, that is why I said in the definition of definitive host, I have to say that it is a host in which the adult stage exists. We come to the second types of parasites, which are protozoa. Protozoa are unicellular microorganisms. This is Gardia parasite. This is a single cell and has two nucleus. In the first lecture, I gave you Leishmania parasite, which is a spindle with a single nucleus and has a single flagell. This is unicellular organisms and can multiply sexually or asexually. This protozoa has no adult stage or larval stage. That is why in the definition of definitive host, we said that you have to set complete definition applied to all parasites. We said it is a host in which adult stage exists or in which sexual multiplication. And we said the intermediate host, it is the host in which the immature stage or larval stage exists, or the host in which asexual multiplication takes place. The third category is arthropod. Arthropods are also parasites. You can see here the mosquito, while taking a blood meal, which is essential for deposition of eggs by mosquito. For the mosquito to complete its life cycle, it has to take a blood meal. Let us start this lecture with helminthes. Helminthes, as I said, it is a multicellular, bilateral, symmetrical, elongated. You see in the previous photo a round worm, but the worm can also be flattened dorsoventrally like a piece of paper. Can be formed of one segment or can be segmented. So, helminthes are divided according to their morphology into two phylum. Platyhelminthes, this is a Latin word, means a plate. A plate is a flattened dorsoventrally. So, we call them flat worms. Platyhelminthes mean flat worms. Or Nematoda, or we call it also Nematihelminthes. The term Nematoda or Nematihelminthes means that they are rounded by Latin language. So they are roundworms. So helminthes can be flatworms or roundworms. If you see here the classification, so helminthology or helminthes can be classified into two phylum, Platyhelminthes and Nematihelminthes or Nematoda. And we have different classes under the phylum. Here we have under Platyhelminthes 4 classes, of which only 2 in fact man, Trematodes and Cystodes. There are other 2 classes which don't infect man, and as we are dealing here in Faculty of Medicine with medical parasitology, we are not going to deal with them. They will be discussed in Faculty of Veterinary Medicine. And Nematoda, the class Nematodes. Phylum Platyhelminthes, we said they are flatworm, flattened dorsoventrally like a piece of paper, and their size varies. Maybe macroscopic, so I can see them by naked eye up to several centimeters, or microscopic to few millimeters, so I have to see them under the microscope. They can take different shapes. They can be leaf-like, they can be pear-shaped, they can be ribbon-like. Alimentary tract is rudimentary or absent. So there is no body cavity. As they are flattened dorsoventris, there is no body cavity. But if they are rounded worm, you are going to find a body cavity. They are usually hermaphrodite, means that they have male and female genital reproductive system inside the same worm. This phylum, as I said previously in the previous slide, has 4 classes. Only 2 of them, Trematode and Cystode, include human parasites. So we are going only to take Trematode and Cystode under phylum Platyhelminthes. Why parasites are classified by this way? You are going to find that all parasites having different shape or different morphology are classified under one category. Parasites classified under one category have the same life cycle. Okay, so if you know how to study parasitology, it will be very easy for you. This is a parasitological classification. Under Trematode, you will find parasites. All of them share the same morphology. All of them has the same life cycle with minor differences. So if you are going to study these differences, so you are going very easy to study parasitology. Under Trematode, you are going to take 3 diseases in module 2. If you study them together, and if you know that they are Trematode, so you are going to recall what are the general character of morphology of Trematode. You will find all of them have the same character. The different life cycle, all the 3 will have the same life cycle. All of them have the general life cycle character of Trematode. Trematode needs water to complete the life cycle, so all these parasitic diseases will need water to complete the life cycle. You will find snail in their life cycle, so in life cycle of the 3 diseases in module 2, you will find them needs a snail to complete the life cycle. So it is very important to study parasitology by categorizing the diseases under classification. So in the second module, you are going to study parasites under Trematode. You have to study them together and to compare between them. Under Cystode, another group of parasitic diseases, another group of parasitic diseases under Nematode, and so on. If you study every disease individually, you will find very difficulty in studying parasitology, but you have to study them and your mind under the classification. You have to remember that this disease is under Trematode, so you are going to recall very easily the morphology and life cycle. We will start today with class Trematode, which belongs to Platyhelminthes, so they are flatworm. General morphology, as I said previously, it is L size. It has different size. Some of them are large, up to 7 centimeters. This is a coin, and this is the worm, a flatworm, flattened dorsoventrally, non-segmented, about 7 centimeters in length. Okay, this is another, and it looks like leaf. It is a leaf-like. If you come to another trematode, it is beer-shaped. Okay, this trematode is seen under the microscope. It is very small, up to 0.1, up to 0.3. We will find it in another photo at the end of the lecture, and under it you will find the measurements. So 0.3 millimeter you can see by naked eye, so it is microscopic. The third trematode you are going to take it Bilharzia. Bilharzia, if you see here the scale, you find this is 1 centimeter. So it can be seen by naked eye, and it is 1 centimeter, but its morphology elongated. And this is the only worm that's not hermaphrodite. It has a separate sex. The thin one is the female, and the thick one is the male. You can tell me it is a round worm. No, it is not a round worm, but it is a flat worm but turned, forming a canal in which it carries the female inside. So it is a trematode. So the general character of trematodes, they are leaf-like, like this one, beer-shaped or elongated worm. Their size vary, maybe macroscopic up to 7 centimeters like this worm, or microscopic like this one, or macroscopic about 1 centimeter. The cuticle means that the outer covering of the worm can be smooth, spiny, or tuberculated. You can't see them under light microscope. You have to magnify it up to several thousands to see. And we can see the scales, the spines, the tubercles under electron microscope. Trematodes are characterizing by having a structure known as suckers. By use of suckers, they attach themselves to the place where they live. For example, this worm is Bilharzia, lives in a blood vessel inside the blood and moves in its direction against the bloodstream. So it has to attach itself to blood vessels so as not to be swept back by bloodstream. They use the suckers. You can see here it has 2 suckers. This is one and this is the second one. This is an oral at the anterior end and this is the ventral sucker below it, used for attachment. Also here at the apical end you can find the oral sucker, and below you can find the ventral sucker. Why I said at least 2 suckers as organ of attachment? Because some trematodes has 3 suckers like this one. It has here oral sucker and another ventral behind it, and a third one will be located called a genital sucker. So all trematode has 2 suckers, but some of them has 3. Has a digestive system, but it is blind, and they are hermaphrodite except schistosoma. Here you can find the male and the female genital system, also here male and female, so both of them are hermaphrodite. But schistosomas is the only exception where sex is separate. The male is a very thin worm, and this is the male. Female, and this is male. Carries a female inside its canal. This is diagrammatic diagram of morphology of trematode. As you see here, it is a flat worm, flattened dorsoventral. Has an oral sucker and has a ventral sucker which is used by the worm for attachment. You can see it has 1 ovary, it has 2 testes, so the worm trematode is hermaphrodite. The digestive system, 2 intestinal ceca which are blind and blindly. Okay, and has uterus and vitelline glands which is essential for oogenesis. All trematode has the same morphology, except the second one, the microscopic one, has a third sucker located here, okay, and the sex is separate in schistosome. All trematodes share the life cycle, so any one of them, if you know the general character of life cycle of all trematodes, so you are going to know the general character of the 3 worms you see them in the previous slide. All trematodes are digenetic. What it means? It means that it has 2 types of reproduction. One sexual in the definitive host and one asexual in the intermediate host. This means that the sexual life cycle occurs in the definitive host where I can find the adult worm. So in trematode you can find the adult worm in the definitive system, in the definitive host, and sexual reproduction occurs in the definitive host. In the intermediate host you are going to find the asexual reproduction, and you are going to find also the larval stage or immature stage. This is unique for trematode. It has 2 developments, sexual and asexual. That is they are digenetic. If I ask you what is meant by digenetic, you are going to say it multiplies sexually in definitive host, 2 types of reproduction, sexually and asexually in the intermediate host. The second character that it lay eggs, okay? And the eggs must reach water. So if you find water in the life cycle of any parasite, so it will be a trematode. The eggs must reach water to complete the life cycle. The egg of trematode are characterized by being operculated. What is meant by operculated? Operculated eggs means that the egg has a lid, a cover. It can open and close. So all eggs of trematodes are operculated except one, which is schistosoma. Okay? This means bilharzia, schistosoma or bilharzia. The eggs can come out from the host mature or immature. You are going in module 2, you are going to take 3 worms. The first is fasciola, or we call trematodes fluke, the liver fluke. The second, the microscopic one, is heterophis, we call it intestinal fluke. And the third one is bilharzia or schistosoma, we call it blood fluke. Why I call liver, intestine, and the blood? Because they live in different habitat. Fasciola in bile duct of liver, heterophis in lumen small intestine, schistosoma in blood vessels. So all will lay eggs, all will lay operculated eggs, except schistosoma. It will have a special character. The eggs can be either mature, immature. In this module, I don't want you to know the names of parasites. All what I want is to know that some eggs are mature and some eggs are immature. But in module 2, when you are going to take fasciola, heterophis, schistosoma, you are going to recall module 1 that they must in life cycle reach water. The eggs are operculated, except schistosoma, it has spine or special character. They may be mature, immature. If I said in this lecture, I have 2 mature and the first one immature, it will be easy for you for module 2 to study the 3 parasitic diseases together by comparing the different character of each stage. So the eggs should be operculated, except in schistosoma, it has a character, a spine, and all eggs of schistosoma of trematode must reach water to complete their life cycle. Okay? In water, the eggs will hatch, liberating a stage known as a miracidium. Since it is present in water, it should be adapted for swimming in water. It has cilia covering, so it will swim in water, searching for their specific snail, which is intermediate host. So, all trematodes will utilize the snails as intermediate host in their life cycle. So, in module 2, the 3 diseases, you are going to find snails in the life cycle of trematode. After snail penetration, the miracidium, the stage, will transform into different stages. We call them different larval stages, aiming to mature from egg to return back to adult stage in the definitive host. So the miracidium will transform into sporocyst. Multiplication occurs inside the snail asexually. So snail will be the intermediate host. It is the host in which asexual multiplication occurs. So multiplication here occurs asexually, or the other definition in which the immature stage or the larval stage exists. Snail, where we find the immature or larval stage, which are sporocysts, which transform into regia, then comes out from the snail cercaria. All the 3 are considered immature stages or larval stages and multiply asexually. One egg, one miracidium, enters the snail, about 250,000 cercaria where comes out. So it is not only one enter and one comes out. No, there is asexual multiplication. So thousands of cercaria will come out as a result of multiplication of the miracidium inside and as a result of penetration of one miracidium, the snail. The cercaria morphology, it looks like the adult. It has a body and It has a tail. The body has the organ of attachment. Like the adult, it has the oral sucker and the ventral sucker, and it has a primitive gut. If you remember in lecture 1, I said that water can be source of transmission of parasitic infection, either by drinking polluted water or unclean water, or while swimming, the parasite will penetrate. So the cercaria will swim in water till it finds its host, but here the fate of cercaria will differ. We have cercaria will directly penetrate host in disease, but other cercaria will, you can see here, cercaria swim in water to penetrate the skin of definitive host, or they insist in second intermediate host or water vegetation. The cercaria can penetrate or search for another water vegetation to secrete around itself a thick wall and transform into cyst. We call it encysted metacercaria. You can find encysted metacercaria on water vegetations, or the cercaria itself can penetrate the skin of another host living in water and transform in the flesh of this host into encysted metacercaria. So what lives in water? Fish, of course, fish. This is an example of life cycle of trematode. You can see here, this is the definitive host, which is man, where the schistosoma, this is example of the worm, you saw it in the first lecture, has a separate sex, we call it schistosoma. It lives here in man. The eggs will come with his excreta. We have different types of schistosoma. Some lives in blood vessels of mesenteric plexus. We call it intestinal schistosomiasis, and causing disease known as schistosomyiasis, intestinal schistosomiasis. And other schistosomes live in the blood vessels of vesicle of urinary bladder and causing urinary schistosomiasis. They lay eggs. That is why we have different eggs, because of different species of schistosome. You see here schistosoma hematobium, you see here schistosoma mansoni, schistosoma japonicum, because we have only one genus schistosoma and we have many species under it. We said that eggs of trematode are operculated, except the egg of schistosoma, which is provided with a spine. You can see here the spine, okay? And it must reach water. The blue one is water. So life cycle of trematode, they must reach water. Inside water, the egg, if mature, will immediately hatch to liberate a ciliated structure known as miracidium. The miracidium should have apical papillae that contain penetration glands to enter the snail. Why here I find 3 snails? Because each species of schistosoma needs a special type of snail. Not all schistosoma can enter any snail. Every species of schistosoma need a certain species of a snail. So we have 3 types of schistosoma, so we have here 3 types of snails. Inside the snail, we said that the miracidium will transform into sporocyte cyst, multiply asexually, then comes out cercaria. The cercaria either penetrates the skin of man, so this to complete the cycle. So diagnostic stage will be the egg that comes out from the definitive host. And the infective stage, if you remember in lecture 1, we said infective stage, it is the stage which infect man. We said the definitive host, it is the host in which the adult stage exists or in which sexual multiplication takes place. And we define intermediate host. It is the host in which the larval stage exists or in which asexual multiplication takes place. We revise together what we said previously in lecture 1. Here you are going to see eggs of trematode. We said that eggs of trematode are operculated, means that it has a lid at the anterior end. With the exception of one of them, schistosoma, it is provided with a spine. So, in module 2, when you are going to take fasciola or heterophysis schistosoma, the 3 diseases, trematode diseases, you are going without difficulty, you are going to recall module 1, Trematode, the general character of trematode, describes the egg of fasciola without thinking. It is an operculated egg, heterophysis is operculated. The only one is provided with the spine is schistosome. We said that eggs must reach water and it will hatch inside water liberating miracidium. We said that should be adapted for swimming in water, so miracidium should be provided with cilia, as you see here. This is the cilia which help her, this miracidium, to move inside water, searching for the snail intermediate host. This is a stained snail with carmine stain in order to visualize for you the cilia, and it has an apical papillae anterior which help the miracidium to penetrate the soft tissue of the snail. These are the different snails of trematode, and as I said previously, that each species of schistosoma or a trematode need a certain genus and species of a snail. This is a snail of schistosoma. Please kindly notice that it is a soft animal inside the snail, and this is the shell. So the miracidium will come and penetrate the soft part of the snail. It is a living structure inside the shell. And this is, in the practical, you are going to see only the shell at the snail. It will enter in the soft tissue of the snail, the miracidium will transform sporocyst redia, and thousands of the cercaria will come out from the snail. For example, this is a Bimoflaria alexandrina. You are not requested in module 1 to tell the name of the snail. You are asked to tell the name in module 2, but here you are going to just say snail of trematode. This is Bimoflaria alexandrina, snail of schistosoma intestinal schistosomyces. This is Apollinus truncatus. This is a snail intermediate host of urinary schistosomyces. This is Lemnia snail, the intermediate host of liver fluke or fasciola, and this is Byronella conica snail, the intermediate host of heterophysis or intestinal fluke. We said the cercaria will come out from the snail, and we said that it has a body and tail, body and tail, body and tail, and it has the general character of trematode because it will become the adult worm. So it is in immature stage, larval stage. It has the oral sucker here, it has a ventral sucker here, it has a digestive system which bifurcate, okay? It has penetration gland because it has to penetrate the skin. We have different types of cercaria according to the type of worm. So cercaria, not all have the same shape. Cercaria has a body and a tail. The tail may be simple, okay? When you take module 2, you are going to find that simple tail cercaria is that of fasciola or liver fluke. The tail can has a membrane around it. We call it a lophocerca cercaria, which is the cercaria of the intestinal fluke, okay, or heterophysis. And the third type is cercaria with biforked tail, which is cercaria of schistosoma. Okay, we call it forcocerca cercaria. The cercaria of trematode, the shape of the tail will differentiate the different types of cercaria from each other. What about the fate of cercaria? We said previously that cercaria of schistosoma, which is biforked cercaria, will directly penetrate the skin while swimming in rivers or canals, okay? While cercaria of the other two trematodes will secrete a cyst wall around itself and will insist. Some of them will insist on water vegetation, and other will penetrate the flesh of creatures living in water, such as fish, and transform inside the flesh of the fish into encysted metacercaria. So the encysted metacercaria can be found in fasciola, we call it a liver fluke, okay, and heterophysis, which is intestinal fluke, but cercaria will not insist in case of schistosoma worm. So when I said general character of trematode, the cercaria of all trematode will insist into insisted metacercaria, except in schistosoma it will not insist. So, if you remember lecture 1, this cercaria as a mode of infection will be by skin penetration, whereas the insisted one, the mode of infection will be by ingestion. These are the 3 examples of parasitic diseases, which are the most 3 important, which you are going to take in lecture 2. They are not requested from you in module 1. We are not going to ask you about them in module 1. But you have to know that they are trematode. If I bring for you in practical exam an image for fasciola worm, you have to identify by being only it is a trematode. I don't ask from you to say it is fasciola worm. The first worm you are going to is fasciola. If you notice here, I said fasciola species because we have different type of fasciola hepatica and fasciola gigantica, and the disease caused by fasciola is called fascioliasis, and it is known as the liver fluke. If you see here, I write the first letter in uppercase letter, and the species, the first letter will be written in a small case letter. This is the second trematode, Heterophis. Why it is called Heterophis? Because if you see Heterophis living, you'll find it changing its shape all the time. The living will change its shape. It is the beer-shaped one that you saw it in the beginning of the lecture. Heterophis, we call it intestinal fluke because it lives in the intestine and is responsible for a disease known as heterophiasis. If you see the disease, we add SIS at the end of the name of the parasite. The third one is a schistosoma species, and we call it schistosomyces, the disease causing it. This is the genus Schistosoma. Under Schistosoma, you will find Schistosoma mansoni, the causative agent of intestinal schistosomyces. We will find Schistosoma japonicum, also cause intestinal schistosomyces, but in the Far East. And the third species, Schistosoma hematobium, which is a causative agent of urinary schistosomyces. As I said, this is the genus and this is the species. The name of the parasite usually written italic. The first letter in the genus should be written in uppercase letter, and the first in the species should be written in lowercase letter, and the word should be written italic. Any organism in parasitology, you will find a genus and species. Also the snail. Snails you will find, for example, you see previous slide, images of Polinus snail. I said Polinus truncatus. Polinus is the genus and truncatus is the species, or Biomuflari alexandrina. Biomuflari is the genus and alexandrina is the species, because there are many species under the same genus. This is the first disease you are going to take in module 2, classified under trematode, fascioliasis or a liver fluke. This is the morphology of the adult worm. We said a trematode, flattened dorsoventrally, has suckers as organ of attachment, okay, and it is one segment, non-segmented. The definitive host is buffaloes, sheep, cattle, goat, okay, and it is a trematode. The eggs will come out from the definitive host, and it must reach water to enter the snail. From the snail, the cercaria will come out to insist on water vegetation, so the mode of infection will be eating insisted metacercaria on water vegetation. If man accidentally enters the cycle, he will act as a definitive host, instead cows of buffaloes, sheep. So it is fasciola, a disease of animal transmissible to man. So I call it fascioliasis is a zoonotic disease. This is revision from lecture 1. And I call fasciola species is accidental parasite because accidentally it enter man and can complete its life cycle. So man act at the same time an accidental host and at the same time definitive host. Why man act as a definitive host? Because the adult stage that exists in cows will exist also in man at the same place in the bile duct of the liver, leading to a disease known as fasciolasis. Okay? Here you are going to see fasciola, you are going to see it in a practical. It is a flat worm up to 7 centimeter, oral sucker, ventral sucker here. It is hermaphrodite, has male and female genital system. Fasciola lives where? It is known as liver fluke because it lives in bile duct of herbivorous animals. It means animals that feed on vegetations. And accidentally or occasionally when man eat unwashed vegetation will have fasciola worm in the bile duct of his liver. The adult worm release the eggs. The eggs will come out to stool. All eggs of trematodes must reach water. All eggs of trematode are operculated, means has a lid at the anterior end, except schistosoma, it has a spine. So when it reaches water, it will mature and the meracidium will come out, swim freely in water. The meracidium then will penetrate the snail. We said that it is a limnia snail. Okay, inside the snail it will multiply asexually and it will transform into sporocyst, regia and cercaria, and comes out one meracidium into the snail, thousands of cercaria will comes out. We said that cercaria has a body and tail. The body has oral sucker, has a ventral sucker, exactly like adult, and the tail will differentiate different cercaria of trematode from each other. We said that cercaria can have a simple tail. We call it a leptocercus cercaria or cercaria with simple tail. The fate of cercaria will be skin penetration or incystation. So cercaria will insist, or fasciola will insist forming incysted metacercaria on which? On water vegetation. This is a lettuce, will insist. So when man eat imperfectly cooked lettuce, we'll take the encysted metacercaria, and the encysted metacercaria will be liberated inside the intestine, transform into adult, and the man get the adult stage in his bile duct. This is simply the life cycle of fasciola, or the first disease, fasciolasis. If I revise with you lecture 1, so this is the adult stage. The definitive host here are animals and accidentally man. So man act as a definitive and accidental host. The eggs of all trematode must reach water. All trematodes utilize snails as intermediate host. The stage which comes is the cercaria. The cercaria either swim freely in water to enter definitive host or insist on water vegetation. The mode of infection is by ingestion. You are going to take the disease fasciolasis in details in module 2. The second trematode is Heterophis heterophis, responsible for a disease known as heterophiasis. We call it intestinal fluke. Heterophis, the definitive host of Heterophis is man. Okay? It lives in a small intestine, that is why you call it intestinal fluke. The eggs will come out with stool, and the eggs is operculated, which is the general character of trematode eggs. The eggs must reach water as all trematode to complete the life cycle. It should utilize intermediate host snails as intermediate host. And inside the snail, the meracidium will transform into sporocyst, regia, and cercaria. The cercaria comes out, thousands of cercaria in water. The cercaria is character of trematode, has a body and tail, but the tail will differentiate different cercaria from each other. It has here a simple tail, so we call it a leptolophocercus cercaria or plurolophocercus cercaria. We said cercaria either swim in water or transform into incysted metacercaria. We said that we have certain type. that will penetrate another living host in water, which is fish. Inside the fish, it will transform into incisive metacercaria. So the mode of infection to man is through eating imperfectly cooked fish or imperfectly salted fish or raw flesh of fish used in preparation of sushi. If you notice here, group of animals beside man, cats, dogs, and fish-eating birds beside man. They are group of animals having the adult stage in their intestine and can complete the life cycle in nature if man not exist. So I call this group of animals beside man, which type of host? I call them reservoir host. That means that they complete or maintain the life cycle of man of heterophis if man is not present. So from this life cycle, I can tell you that the definitive host of heterophis is man. The reservoir, dogs, cats, fish-eating birds. The stage which comes out from man is the diagnostic stage where I examine the stool of infected person. I will find the egg. The egg is characterized like all eggs of trematode by being operculated. The egg, like all trematodes, must reach water, must use a snail. The snail is the intermediate host because larval stage exists, asexual multiplication takes place. The cercaria comes out, it has a membrane around tail, it must insist in another living host. So the insisted metacercaria inside the flesh of fish. Since it is larval stage, so I call it a second intermediate host. So the snail for heterophis is the first, while the fish is the second because it harbors the intermediate in the larval stage of heterophis. I call it second intermediate host. Eating imperfectly cooked fish, and this is the mode of infection of heterophis eutrophis. We'll come now to see images. This is adult heterophis. I said that trematode, it is a flat, dorsoventrally, non-segmented, has oral sucker here, a ventral sucker. If you remember lecture 1, we said that all trematode has 2 suckers at least. And I said that there is one has a third sucker in this area, we call it a genital sucker. All trematode are hermaphrodites, so I am going to find here male and female genital system. I said the size may be up to several centimeters or microscopic. Look here to the scale, it is 2 millimeters. So I can't see heterophis except under the microscope. So it's a flat worm, has suckers, non-segmented, hermaphrodite, microscopic. It lives in a small intestine, so the eggs will come out with the stool. All eggs of trematode, we said, operculated. You can see here the operculum. The egg must, like all trematodes, last reach water. In water, the miracidium will be liberated to penetrate the intermediate host, which is Pyrenella conica. Since heterophis utilizes 2 intermediate hosts, in which here I find the larval stage and cercaria comes out, and in which here asexual multiplication takes place, I said this is the first intermediate host because I will find the fish as a second. Which fish heterophis utilizes? The buri and bolti. That is why you can link our information together. Heterophis is present in Rashid here in Alexandria because it is an area where you can find buri and bolti fish. Inside the flesh of bolti and buri, you will find the encysted metacercaria. So buri and bolti are the second intermediate host. Encysted metacercaria is a larval stage. Since it is present in a living organism, so I call it intermediate host. So the only trematode that has 2 intermediate hosts, heterophis, had the first is the snail like all trematode, and the second is buri and bolti fish. The mode of infection is through eating imperfectly cooked fish or under-salted fish. This is fisik, okay? This is imperfectly cooked fish, grilled fish, and this is sushi if people now use flesh of balti fish. Now it is prepared in the market as a fillet, can be used in preparation of sushi at home. The third disease, trematode, is a schistosomiasis caused by schistosoma species. We said schistosoma is a blood fluke because it lives in fine capillaries blood vessels. We have different types, intestinal and urinary. The intestinal lives in blood vessels of mesenteric plexus. We have two species, schistosoma mansoni and schistosoma japonicum, responsible for intestinal schistosomiasis. And we have a third species, schistosoma hematobium, lives in blood vessels of urinary bladder. It's a vesical blood vessels. Schistosomiasis, as you see here, if you remember, it's the cercaria, does not insist, and it penetrates through swimming in water, penetrates the skin. You remember we said previously while saying the life cycle of fasciola and heterophis, and like all trematode, schistosoma utilizes snail as intermediate host and must reach water to complete the life cycle. Schistosoma is exception because it has separate sex, not hermaphrodite. Schistosoma is exception because the egg does not have operculum, it has a spine. This is schistosoma worm, as I said previously. It has separate sex. This is the female inside canal of male. It has oral sucker here and it has ventral sucker below. You are going to see in the practical schistosoma worm. Don't worry, you are going to see it in details. It lives in blood vessels and through the fine capillaries supplying the intestine or urinary bladder, the eggs will come out and penetrate the blood vessel to enter into stool or urine. To intestine comes out with stool, or in urinary bladder to comes out with urine. As you see, eggs of schistosoma has a spine, a character. Also, each species of schistosoma, the schistosoma mansoni has egg with lateral spine, but schistosoma hematobium has egg with terminal spine. So eggs of schistosoma are not operculated, but has a characteristic feature that is spine. The eggs must reach water to reach its snail intermediate host, and as I said previously, each species needs a special snail, and the snail also has a genus and species. This is Biomphalalia alexandrina, the intermediate host of schistosoma mansoni, responsible for intestinal schistosomiasis, and this is Polinus truncatus, the genus Polinus truncatus species, which is responsible for urinary schistosomiasis. Inside the snail, the miracidium will enter, multiplied sporocyst. There is no radial stage, it's not important here. And the cercaria comes out. The cercaria, like all trematode, has a body and has a tail, and the tail is characteristic by forked tail. We said that this cercaria will swim freely in water until it finds host. In man, this is a stained cercaria. You can see it with carmine stain. This is the head and this is the tail. Forked tail. This is living one and this is stained one. Until it finds man. This is a photo by scanning electron microscope. You can see here the cercaria will penetrate the skin of man while swimming. This part will enter and it will leave the tail in water. Okay, but the mode of infection is through skin penetration. From this picture you can conclude that the mode of infection is through skin penetration while swimming or while washing kitchen towels inside water. Here you can find the adult worm living in man, which is considered definitive host. The eggs, like all trematode, differ in having spine. The eggs, like all trematode, must reach water. Like all trematode, it utilizes snails as intermediate host. The cercaria comes out from a snail by forked tail. Mode of infection is by skin penetration. So the portal of entry, penetration of skin. Here I said again, it is an active skin penetration, contrary to mosquito fly where it introduced malaria passively, and the mode of infection through swimming or contact with polluted water. The disease affect the liver. If you remember in the first lecture, we said that one of the pathogenesis of parasitic disease is the stimulation of host immune response. Here the eggs of schistosoma can be swept by bloodstream and go to liver. Inside the liver, there will be stimulation of host immune response because it is a foreign body. Each egg will be surrounded by granulomatous reaction. As a result, granuloma will be formed. As a result, hepatomegaly will occur. And as it is connected to the portal circulation, the spleen will be enlarged. As a result, hepatic dysfunction, ascites, and you will have hepatosplenomegaly, huge hepatosplenomegaly, and ascites. These are patients suffering from intestinal schistosomiasis. I give you an idea about the 3 diseases. You are not requested in exam of module 1 to know these details, or what you have to know that when I bring you this photo in exam, you have to say that it is a trematode. When I bring this egg, you said it is an egg of trematode. You don't have to specify to which parasite it belongs. You have to say that this is a snail and it acts as intermediate host of trematode. When I bring you this picture, you have to say this is only a surcarrier of trematode. I don't want from you to specify it is for schistosoma. But I give you a brief idea to help you to study well the 3 diseases you are going to take in module 2 under trematode. Please remember to study them together. After you take the 3 diseases of module 2, you have to study them in sort of comparison, because this will help you easily to study parasitological diseases. In the subsequent lectures, you are going to take other parasites belonging to another class, Trematoda and Nematoda. You will find that all cystode diseases will have the same character, the same for all nematodes and so on. So it will be easy to study parasitic diseases in groups, not singly or individually, so not to be confused. I hope you understand my lecture, and if you have any question, please come and ask. Don't hesitate to come to my office and ask me. You are more than welcome, and I would like to thank you for your attention. Thank you.